Hi, my name is Lucia Seale, and I'm an assistant researcher at the PBRC University of Hawaii at Manoa. Today I'll present my research on selenium recycling in the brown adipose tissue. Selenium is a micronutrient essential to life, found in various foods such as seafood and Brazil nuts. Americans in general have a high intake of selenium. Selenium is found in our bodies as part of selenoproteins, which act mainly as antioxidants and regulators of thyroid hormone activation. The pathway shown here has been the focus of my career since graduate school, particularly the actions of the enzyme Scly in orange. Scly bridges selenoprotein synthesis and degradation. Therefore, it regulates selenium recycling in cells. We developed the Scly knockout mouse, and this mouse model develops obesity when exposed to a selenium-deficient diet, making it a selenium-dependent obesity model. They have an enlarged brown adipose tissue, and we decided to turn our focus on the role of selenium and Scly in this organ. The brown fat is the main site of adaptive thermogenesis, a mechanism involved in the regulation of energy expenditure and dysregulated by diet-induced obesity. Hallmarks of adaptive thermogenesis are an upregulation of proteins UCP1, PGC1 alpha, and selenoproteins type 2 deiodinase, or DIO2. The essentiality of DIO2 for brown fat thermogenesis was the focus of my first manuscript. My objective with this work is to determine the role of Scly in brown fat thermogenesis. This is the focus also of an R1 submitted last June and planned to be funded by the NIDDK. To understand this, we have fed selenium-deficient diets to Scly knockout mouse, exposed them to 4 degrees Celsius, and assessed for energy expenditure and selenium parameters in the brown fat. We observed larger lipid droplets in the brown fat of Scly knockout mice, altering its morphology, and a localized selenium deficiency. We also observed lower core body temperature after 6 to 9 hours into cold, suggestive of a temporary impairment in thermoregulatory capacity. Moreover, we found a downregulation of thermogenesis marker UCP1. We also found that selenoproteins SelenoW and thermogenesis marker DIO2 were decreased in these mice. Our preliminary conclusion is that Scly controls selenium levels in brown fat, impairing the synthesis of DIO2 and SelenoW, which transiently affects response to adaptive thermogenesis. I am grateful for the contribution of several people in our lab and collaborators, and above all, I'm grateful for the funding agencies that supported our studies. Thank you.